Okay, welcome back to the part 3 of today's lecture. Now, the culture medium and the media reagents for mammalian cell culture are relatively different than bacterial and plant cell culture. You could see when we talk about mammalian cell culture, there are three different types of general classes of media. One is known as basal media, and the other one is known as reduced serum media. The third and the last one is serum-free media. Now, as we know, the culture medium is the combination of the ingredients which will support and maintain the growth by providing all the essential nutrients. For example, in case of mammalian cell culture, we have to provide both essential and non-essential type of amino acids, which means all 20 standard amino acids. When we talk about bacteria and yeast, we have to provide some amino acids in the medium, but if you do not provide amino acids in the medium, they still have the capacity to synthesize. Bacteria have this capacity to synthesize almost all amino acids. But in case of cell cultures of mammals, mammalian cell culture, we need to provide all amino acids in the growth medium. Again, just like in bacterial and fungal cell culture, the choice of the culture medium is dependent on the requirements of the cell culture as well as the purpose of the cells, why we are culturing the cells, which kind of investigations or which kind of product synthesis we are going. Because I can tell you at this point that even some of eukaryotic cells could be used for the synthesis of recombinant proteins. Insect cells are used. Yeast is already a eukaryote, but it's a microbe, so that's understood. But higher eukaryote cells could be used. One of the classical example is using tobacco plants, using tobacco mosaic virus for the synthesis of a number of vaccines, because it's very easy to produce vaccines in plants in higher amounts and easy to purify. Similarly, in other higher mammals cells, you could clone the gene and you can use them for production of a number of recombinant proteins. Not only bacteria, not only yeast, but plant cells, as well as insect cells, as well as higher eukaryotic mammal cells. Talking about the culture medium for mammalian cell culture, the basal medium is a medium that contains essentially all amino acids, vitamins, inorganic salts, the trace elements, which also we call it the micronutrients, which are very important for a number of enzymes to be as their cofactors. But it does not contain, the only thing that is missing in the basal medium is the growth supplement. For example, serum we add in growth medium in mammalian cell cultures. In the growth media, we always add sera, animal sera. Animal sera are actually rich in a number of growth hormones, phosphates, and other compounds that are really important, organic compounds that are really important for the medium, for the thriving, for the maintenance and growth and differentiation of these cells. But in basal medium, there would be nothing. For example, DMEM, RMPI, could be with or without glutamine. These are our very common basal medium classes of cell culture media for mammalian cells. Then second type is reduced serum. Reduced serum medium is just like the basic basal medium, but there is a little amount of serum added. So you have the basal medium formulation enriched with nutrients and animal-derived factors. Like you do not need a high amount of serum because it already has some formulations from the animal-derived factors. You still add serum, but not a lot of serum. Then you can have a completely serum-free medium. Now, when you have a completely serum-free medium, it will go here. So when we talk about complex and defined medium, serum is a complex medium. We do not know exactly what is inside and in what proportion. But when we talk about serum-free medium, then we are talking about defined medium, because here we are preparing a medium in which all the appropriate nutritional and hormonal formulations are actually included in it, and we know about it. And this is very important for the primary cell cultures, as for example, Chinese hamster ovary, hybridoma, some cancer cell lines. These are all different mortal and immortal cell lines which are cultured through serum-free medium. The major advantage of this medium is that you can make a medium selective for specific cell types. So as I discussed, this is a selective growth medium. You are working with mammalian cells X, let's suppose, and you know that these mammalian cells X require these A, B, C, D, E factors. So you would go for a medium which has only A, B, C, D factors. You would not go for something, a complex growth medium where you have A, B, C, D, F, G, H, X, Y, Z. You would say, okay, there are so many nutrients extra, but we don't care. No, you go for the selective specific medium and only the ingredients that are needed for your cells, so that you can draw your conclusions or you can produce your proteins, recombinant proteins, the way you want. These are some common culture mediums available and produced by a number of biotech and molecular biology companies, and you can purchase them. These days, nobody synthesizes their own growth medium, not even for microbiology purposes, which is rather simpler in comparison with mammalian cell culture. Minimum essential medium, GMEM, EMEM, DMEM, medium 199, the basal eagle medium, the HAMS F10 medium, F12 medium, RPMI medium. These are just media which are named because of their composition or because of a company, like McCoy's 5A. It's just a name. We do not know what is a McCoy unless we go and check the ingredients. You can purchase them from the catalog and from different companies. You can use them. They're ready to use. All of these come in sterile packages. You just have to keep them sterile once open. Okay. Now, the process of culturing mammalian cells. There's a difference between mammalian cells and plant cells, as well as bacterial and yeast cells. Plant cells and microbial cells, when I say microbial cells, it includes both bacteria and fungi. They can be cultured on solid or semi-solid surfaces as well as suspended. They don't care. If you culture them on an agar plate, they will very nicely get plated out on the on the surface of the agar plates and grow. And you are not happy with it. You want to culture them in a liquid medium in Erlenmeyer flasks by continuous shaking in a shaking incubator. And you will see they will be happily, okay, they will say, okay, why, there's no problem. They will culture and they will grow in it, which means the same is the case with plant cells. We discussed it in plant tissue culture. You can use agar plates, agar slants, as well as suspended cultures. But in case of mammalian cells, this is not the case. These cells, they are anchorage dependent cells. Anchorage dependent cells means they always need a surface to adhere to. Normal mammalian cells cannot culture, they cannot grow under suspension conditions. So if you take, let's suppose, a muscle cell from a hamster, and you want to culture it in an Erlenmeyer flasks in a suspended form, they will never culture. It's not possible. They need to have a scaffold to stick to something, and only then they can culture. So we call them anchorage dependent cells. And they will not grow unless they're attached to a surface. Even inside our body, all these cells, they are anchoring to something, either part of the bone, a part of the tissue, the part of the other cells. For example, one of the example in this case is your intestines. So the cells in your intestines are all epithelial cells. It's like a big long sheet of epithelial cells which is rolled into a tube. But these cells, they are connected to each other by tight junctions. And a tight junction is a protein, is made by proteins. Half of the protein is made by one cell and half of the protein is made by the other cells, and they interact and they stick together. Similarly is the case for almost all other cells. There is a small exception even in case of mammalian cells. Now I'm talking about normal mammalian cells. Normal mammalian cells, majority of them are anchorage dependent. Some of them are not anchorage dependent, and it will come to your mind when you think about a human body or any higher mammal. There are cells which are in your body and they're flowing around, suspended form. Which cells are those? We will come to see it in the next slide. Sometimes these anchorage dependent cells, cells that are not flowing around, but they are rather sticking to something, and even then they are growing, only then they are growing. They can also undergo apoptosis. Apoptosis is the programmed cell death if they are not attached to a cell. So you have a muscle cell, you want to culture it in a suspension liquid. It might be the cell triggers apoptosis and it dies. It never grows. Mammalian cells, they usually make a monolayer. So they make a flat layer eventually over the entire surface of the medium. Once the medium or what the monolayer covers the whole culture vessel, you have to shift them to another source, another petri plate or another culture vessel, because they cannot grow once the monolayer is completed. This is not the case in plant cells as well as in bacterial cells. Now, as I said, some of the cells are flowing around in your body. These are the blood cells. I mean, blood cells are cells, white blood cells and red blood cells. They can actually, you can culture them in suspension culture. And we call them non-anchorage dependent cells. When we talk about mammalian cells, we already had a distinction between anchorage dependent cells and anchorage independent cells. With the blood cells are anchorage independent, the rest of the majority of the cells are anchorage dependent cells. Again, the mammalian growth medium, it contains, this is something which already discussed, there are pH indicators. I think this is not necessary. Now, this is very important. Primary cell cultures and secondary, not secondary, but we call them with another name, mortal and immortal cell lines.